All right, welcome to Chapter 8 on genetics. So genetics is the study of genes. Before we get into genetics, we're going to talk about a few different terms, definitions that you should know so that we're kind of on the same page of what we're talking about. So the genome is all the genetic information for that cell. So it's going to include all of the genes. One gene is a segment of DNA that encodes for a functional product. Most of the time, this functional product is going to be a protein. A chromosome is the structure that contains those genes. Remember, in bacteria and in prokaryotes, we're only going to have one circular chromosome. So that one circular chromosome is going to contain all of the genes. And again, each gene is encoding for one protein. The genetic code, that is how the nucleotide sequence is converted to amino acids. So think of it as like any code. It's like translating from one language into another. It's a translation code. So we're translating from nucleotide language to amino acid language. So that's what the genetic code is. And then lastly, we have genotype and phenotype. So a genotype is the genetic makeup of an organism, and the phenotype is how that is expressed. So for example, your genotype would be the genes that you have, say, for eye color. The phenotype would be how that's expressed. So if you have brown eyes, your phenotype would be brown eyes. That's the physical expression of the genotype. And the genotype would be genes that are encoding for the brown eyes. So again, bacterial DNA, we only have one singular circular chromosome in the nucleoid region. So in this picture, that's going to be this kind of fainter purple. I think it's showing that it's deeper in the cell. That's just one circular chromosome. Now, in addition to the circular chromosome, there can also be plasmid DNAs. So they're also circular. They're much, much smaller. And again, they're optional. So the cell may have one, it may have several, it may have none. And the genetic information carried on those plasmids is also optional. So it's not going to be coding for genes that are necessary for basic survival, but it can be carrying genes that add to virulence to make that organism maybe potentially more pathogenic. For example, like antibiotic resistance is often carried on plasmids. All right, so this slide is kind of like a summary slide of everything that we're going to be talking about. So there's three different methods of genetic information flow. The first that we're going to talk about very briefly is replication here. So replication is the flow of genetic information from one generation to the next generation. So we're going to talk about that first. Then the majority of this presentation is going to be on expression or gene expression, and that is using that information that's carried in the DNA for carrying out the cell's functions. So that's going to involve transcription and translation to form these proteins that's carrying out the function of the cell. So expression is within that cell and carrying out those functions. And then lastly, we're going to finish up by talking about recombination. This is the flow of information from one cell to another cell. So they're sharing genetic information. All right, so starting off with replication. So replication, again, is we're going to be taking one chromosome and making two chromosomes, and this is going to be carried out during cellular division. So what's going to happen? This is, of course, bacterial chromosome because it's circular. So we still have two strands, though. And DNA replication is going to start at the origin of replication. Not a lot of creativity on that name, right? It's where it starts. what's going to happen is those two strands are going to separate, and each of those separated strands is now going to serve as the template, or a DNA template. And so it's going to form a new strand on each of those old strands. So here we have, they're representing the darker purple as an old strand, and a lighter purple as the new strand that's being built for each of these new chromosomes. That's what we call semi-conservative. So that's meaning part of each new chromosome is going to be one of the old strands and a new strand. The old strand being used as a template to build the new strand of DNA. So that's going to start again at the origin of replication, going to continue around the entire chromosome till it reaches over here, the termination. Also, let me point out that this is called the vertical gene transfer. That's the flow of information from one generation of cells to the next generation of cells. So that's why it's a vertical gene transfer. Make sure you know that key term. All right, moving on to gene expression, which is going to be the majority of Chapter 8. So this slide is just reminding you that DNA, making more DNA is replication. We just talked about that. The enzyme that carries that out, the primary enzyme, is DNA polymerase. I do want you to know that. Now, gene expression is this process here. So it's the process of turning DNA into RNA into protein. And proteins, of course, carry out the traits. So this has two different processes. The process of converting DNA into RNA is called transcription. The main enzyme that carries this out is RNA polymerase, because it's making RNA, right? You can remember this because it's the same language. DNA and RNA both use nucleotides. So it's just rewriting it down. It's transcribing. So that's transcription. Whereas converting RNA to protein, or the process of translation, is changing languages. So RNA, again, is nucleotides. For proteins, the language is amino acids. So now we need to change languages. So it's being translated. So that's how you can keep those two processes straight. Translation, now we're converting it to amino acids, which of course is the base for proteins. This process is carried out by ribozymes. And yeah, so that's overall for gene expression. So let's go through those two processes. First of all, transcription. Again, transcription is carried out by RNA polymerase. It's going to bind to the promoter sequence. Now, really quickly, remember with DNA replication, it's going to replicate the entire genome. It's going to replicate all the genes because we want to make sure that each of the daughter cells gets a complete set, right? So we're making a new set of DNA. Where here in transcription, we're not going to transcribe all of the genes. We're only going to transcribe one gene at a time. This is really important because we usually don't want all of the genes, especially in a multicellular organism such as ourselves. Most of our cells are only going to transcribe a very small number of the genes in that cell. This is important for conserving energy because making proteins takes a lot of energy. So there's no point in making proteins that the cell doesn't need. So transcription is only going to transcribe genes that are needed at that time. The way the cell controls this is it's going to start transcription at the start of a new gene, the gene that's needed. And that's called the promoter. So here, RNA polymerase is going to bind to the promoter sequence. And then it's going to transcribe down, making a new RNA strand that is using one of the DNA strands as a template, forming the RNA until it reaches the terminator. And then it's going to release. We have our new RNA strand for that gene. Next, translation is now going to use that messenger RNA that we just made. And now this is going to serve as the template for making amino acid sequence. Let me just bring all these up. Okay. So there's several players here in translation. I already mentioned the mRNA. We also have the ribosome. That's the enzyme that's going to carry this out. We have tRNAs. So tRNAs are going to match what we have here is an anticodon on one end of the tRNA. That is going to match the codon on the mRNA. So here's the mRNA. Every 3 nucleotides is a codon. Okay, so here we have AUG, that's one codon. CGC, that's the next codon, codon 2. And those are going to match the anticodon on the tRNA. So we see U matches with A matches with U, C matches with G, right? So this is the anticodon on one side of the tRNA, and that's going to correspond with the appropriate amino acid on the other side of the tRNA. So it's through this process of tRNAs going to bring in, so we're going to get the right tRNA with the right codon, tRNA with the right codon is going to bring in the correct amino acid. So that's how the amino acids get put in their correct order. So that's the process of translation. All right, I think we went through that. So this is the genetic code. So I do want you guys to be able to use this chart. So all you need to do is if I give you the order of DNA, be able to put that into mRNA. So, you know, A matches with T, C matches with G, right? I always remember that, like the C's and the G's kind of look the same. They match together. They're kind of that same curvy shape. Don't forget in mRNA, hopefully you've learned this in other classes. Once you get to mRNA, then we don't have T's anymore. That's replaced with U's. So that's why you just see U's here in this chart. So if that's too many steps for you, you can just convert it to A, T, C's, and G's, and then cross out all the T's and replace them with U's, and that's going to be your mRNA. Then you're going to make codons, so break them up by every 3, and then use each codon to find the matching amino acid. So this is the first position, second position, third position. So for example, if we have ACA, you're going to come over here to A, so you know it's in this row. Then you're going to come to C, so then you know it's in this box. This is AC, ACA is this amino acid here. Okay. But note that there's actually 4 different codons. These are going to be the codons because you're looking directly at the mRNA sequence here. Okay, so use your codons when using this chart. But you see there's actually 4 codons here that encode for the same amino acid. And we call that the degeneracy of the genetic code. So it's like it's redundant, right? Several of the codons will encode for the same amino acid. This is kind of convenient because we can have some minor mutations that won't be that detrimental, because if the gene originally was ACA and then it mutates to ACG, well, it doesn't matter because the amino acid is still going to be the same. So you're still going to get the same protein. All right. So this is specific for prokaryotes. In a eukaryote, we produce the mRNA, and that mRNA has to leave the nucleus, right? So transcription is going to happen in the nucleus. mRNA then has to leave the nucleus and go out into the cytoplasm, where it will then be transcribed. But in the prokaryotes, we don't have a nucleus. So both processes can happen simultaneously. So we can have transcription producing this red mRNA, and then ribosomes can come along and bind to the mRNA and start producing proteins before that mRNA is even completely finished. So we call that a polyribosome. So we have multiple ribosomes along the mRNA, which can happen as transcription is still occurring. We're going to go into prokaryotic gene expression. We're going to take a break and then we're going to pick up on that, and that's going to be a good chunk of chapter 8. But before we get into prokaryotes, I'm going to finish this part up by talking about eukaryotic gene expression, kind of how we're going to touch on a few of the ways that that happens. Before we do that, gene expression in itself is, or a regulation of gene expression is the process of determining which genes will be turned on and which will be turned off. When we say a gene is turned on, it means that it's being transcribed and translated into a protein, okay? And again, as I already discussed, we're not going to have all the genes on all the time. Actually, probably most genes are going to be turned off. But the genes that are turned on through the regulation of gene expression. In a eukaryotic cell, we have a few different ways that this can happen. One is that we can methylate the histones. This is only going to happen in eukaryotic cells because only in eukaryotic cells do we have histones, right? But the histones, remember, are the proteins that the DNA is wrapped around. Well, if we methylate them, that causes the DNA to wrap around those histones even tighter. And that's going to convert it into this heterochromatin. So the DNA in that region is wound very tightly so that the genes cannot be the DNA cannot be transcribed. If it's non-methylated, instead it's acetylated, which is represented by the blue here, then it's going to be much looser. So see how we have DNA is kind of looser here? Because it's looser, then the RNA polymerase can now bind and transcribe that DNA. Another way that gene expression can be regulated in eukaryotes is through transcription factors. So the presence or absence of those transcription factors can control whether a transcription will occur for that gene. Transcription factors being needed for transcription. And lastly, we also have mRNA splicing in eukaryotes. Again, this is specific for eukaryotes because this is going to be occurring in the nucleus. So the mRNA will be processed and modified before it leaves the nucleus. And that's not going to happen in prokaryotes because we don't have a nucleus. So a couple of things that happen in eukaryotes, we're going to add a 5' cap, add a poly A tail. These two things help to increase the life of mRNA, helps to protect it. It also is going to help the cell recognize its own mRNA as opposed to, say, viral mRNA. And then it can also splice out some parts of the mRNA. So this is where we can kind of change that mRNA a little bit. So we can remove these introns. I remember it's like the introns they're just in there. They're not really needed, they're just in there. So that's what's removed. All right. So that wraps up eukaryotic gene expression. We're going to take a break and pick back up talking about prokaryotic gene expression and operons. I'll see you guys soon.